Now, the disease of Alzheimer's has what we call selective vulnerability. Only some neurons are involved, others are not. And these involve, for example, these what we call the projection neurons. Neurons in one lobe of the brain that project to another, or to one gyrus to another. And there's other neurons that are nearby that just, it's like a computer, I guess. There are little neurons nearby that modify the behavior of the big neurons that are connecting. And the end of the axon, which connects to the next neuron, there is what's called a synapse. And that's important because it's thought that that's where the disease may begin and works back to the cell body. Now, these are intracellularly within the neuron only, and only certain neurons, there are changes in proteins in it. And they collect together in an abnormal way, and this sort of clogs up the cell, and including the processes that connect it to other cells. And these are present here, what are called neuropil threads. And this is an intracellular abnormal protein that tends to fold abnormally, and that's very important in the disease process. And many people are working on that, using that as a marker within the brain. And they form what are called neurofibrillary tangles. That's an abnormal aggregation of a normal, what had been a normal protein, but which is modified, not genetically necessarily, though there may be small point mutations, but it can be modified by what's called post-genetically, post-translationally. And that's one area where we can just use those markers to make our diagnosis using antibodies or other tools to look at these nerve cells and say that's what we see in Alzheimer's, those changes, and I'll show you those. And there's this is intracellularly. Extracellularly, we have amyloid. I think people who know about Alzheimer's know about plaques and tangles. Those are the abnormal proteins that are in the brain. These are used for imaging. There's one called the Pittsburgh compound, in which these abnormal protein aggregates can be detected by antibody that binds specifically when inserted into the spinal fluid, can bind, or it can bind to these abnormal aggregates of protein, and that's one way when they're present that people make a diagnosis. But that happens pretty far along the course of the disease. So here's how these look, these abnormal proteins. Here's the one I showed you, the neurofibrillary tangle. Here it is. It lights up here using a histochemical marker. And here it's inside these long, big neurons. And it's outside, around, and it's associated with tangles within the neurites, which are not the cell body, but around it, outside, extracellularly, is the amyloid protein. So these come together, so it's called the plaques and the tangles. And we use these for our diagnosis. We now use antibodies directly against parts of each of these. And the tau protein, which forms the tangles, we see here an electron microscopy of a nerve cell, and here's these aggregated proteins sitting here. They're just plugging up the cell. They're not normal, and they have abnormal modifications as well. That's one marker, and this is at higher magnification. It's a helical set of filaments. It's a bifilar array here, and it's just an enlargement of this. And these were first seen many years ago, and they're abnormal in this disease, and maybe they're in some other diseases, not entirely just in this, in Alzheimer's disease. But it is in the distribution that is specific for Alzheimer's, and it's present in the disease, and it affects the axons, the neural processes. It sort of plugs them up, and that's important because they probably aren't functioning. Now, here's another part of the brain that's the most important part and the earliest part to be involved. These are in what's called the hippocampus. And I showed you the sensory regions of the brain topically. You could see, couldn't see it, but you have to take my word where it's shrunken. There are abnormal cells in there with these plaques and tangles. But the first place to be involved is in another area, which is, it is affected most earliest in the disease process. So first you can see these, and it's called the hippocampus. So it is developmentally or phylogenetically one of the earliest parts of the brain to be involved, and not in other species, but at least in the human. And this is where all this sensory input comes into these cells here, and then goes into here. And this is where, and it's various kinds of sensory. It's the somatosensory and perhaps hearing and touch and so forth that shift into these areas. And that's where memory is encoded. This is the first place to be involved. And so there's a stereotypic pattern of involvement by these changes in the brain. And here's the first one. Now, if we could, and so this is what the tests for memory are directed, because this is what is involved earliest. This is a cartoon of it. These are cells in these regions. You don't have to worry which region. I do, though. And this is the part here in the hippocampus called the CA1 region. And this is where memory is encoded. But this area here has all these abnormal proteins in it here, but not in here, and not much over here. So all this input comes in over here, crosses over, goes through here in a little cascade, and that's how memories are made. It's not very poetic. That's the truth. That's how it is. Okay. So that's important. And if we can see this disease at this point, then you know it's really early. If you see it out in the other regions of what we call the cortex of the brain, that means a lot of damage is already underway. So if you see it just here, you hope it could be reversible. You hope to be able to reduce these aggregates of abnormal proteins as well. And that's some of the goals. Here's the pattern we call Brock staging. Somebody named Brock, who is in Germany, worked out this pattern. So very early in the course of the disease, here's normal brain. Here's the side of the brain I showed you. Here's, you turn it around, there's where the hippocampus is. This is flipped too. But this goes from here, then worse here, and a little bit up here. And this is where the first memories are encoded. And then over here, it's really bad. And we stage the disease this way. So we can know how bad it is at this point. And probably at this point, that we have no ways to reverse it at this point. But we know by the neuropsych testing and by the radiological testing that it's that far along. And that would be go along with the staging of the disease and correlate with the behavioral dementia component. Here's in Alzheimer's the neuritic plaques. Here the amyloid is present. That's toxic. And it induces the nerve cell to sprout and make little short circuits in here. And so that becomes a hallmark just of abnormal brain. And so our hypothesis is, and others as well, believe this, Alzheimer's disease begins at the synapse, and that is ground zero. So it starts at the synapse, where one neuron will touch the next one. And there's a lot of connections in the hippocampus as well, where it goes one to the next. And it's thought to spread then out to the other parts of the brain, away from where it first starts. And that's how the disease, and it goes on over a long time. And there are theories how that happens, but I'm not going to get into that. I'm not quite sure they're well proven yet. And so we looked at some of this and defined and select a patient population by their neuropsych and neuropath criteria. We wanted to look and follow the dictum of the NIA. After all, they have the money to give. And we wanted to first look at the synapse. Do we find anything abnormal in the synapse? And what we were looking for was to enrich biochemically the brain. And we have brain that we freeze. When the patient comes in, we dissect the areas that are early in the course and later in the course. And then we freeze tissue, and we can dissect these regions that occur earliest on the brain, and we biochemically sort the synaptic terminals. There's a way of biochemically doing this using what we call synaptoneurosome preparations. There's just the end of the synapse and where it connects to the next neuron that we're able to spin this out and look at what's in there. If it begins there, we should see what molecules are affected in there and what other genes are affected. And then this is, we determine the protein in these, and we look at what we call the messenger RNA enrichment. This is actually a reflection of what the DNA is doing. And if there are small changes in the DNA, it should be reflected in the messenger RNA. The order is DNA to mRNA copying it to protein. And we can look at the proteins and the mRNA in this. And there's a way one does this. We did this work in around 2009. And we can do, and we want to look at it early. What are the early changes we can map out? More than what we all know about, there's a few mutations in the processing of the A beta protein or peptide. And they used what we used was a chip which hybridizes to what's called the mRNA to the DNA, or to, And we use assays that produce what we call in vitro translation and bioinformatics to assess function at the synapse. And then we can find interesting genes. You don't have to know all this, just see the process and how we're doing this. And it's displayed on a microarray, and we're able to see. Now, I'll just give you a little bit so you see how we're approaching this molecularly. Each one of these is from a patient, and this is based on the amount of dementia they have. There's tests that we can test global function, and this is the control. Each one of these is of a patient further along in the course of Alzheimer's and confirmed by the neuropathology and clinically. And this is the earliest AD, the earliest Alzheimer's over here. And this is how the genes, certain genes, each one of these is a gene and it's mRNA expression. And you can see over here, this one was almost normal, and this green was a very low expression. Over here, it's a high expression, and we can detect that. And there's bioinformatics that help us. And so we're able to see with each patient that is more and more in the Alzheimer early dementia range, that these are changing. And here the expression is more over here, probably for the same gene. In a control, it's less. That's some of the approaches that we can take on this. And these are mathematically covered over here. And they're sorted, so we have them sorting here, a large pool, then we do more rigorous sorting of these genes.